Hello, this is Rose, and today I'm gonna go through my potato ethylation and DE-etoliation. And so here we go. Okay, these shoots are the first things that are noticeable after you get the potatoes after the dark. And the first question that comes to mind is why are they here and why are potatoes better off to making these shoots? So here's the reason. These plants, as you can see, are ghostly pale, bare leaves that are unexpected, and lack elongated roots. These morphological adaptations for growing in darkness, collectively referred to as ethylation, makes sense if we consider that a young potato plant in nature usually encounters continuous darkness when sprouting underground. Expanded leaves would be hindrance. A potato plant growing in the dark allocates as much energy as possible to the elongation of stems. The ethylation response is one example of how plants' morphology and physiology are turned to its variable surroundings by complex interactions between environmental and internal signals. When a shoot reaches the sunlight, the plant undergoes profound changes. Since signals are first detected by receptors, proteins that undergo conformational changes in response to a specific stimulus. The receptor involved in the ethylation is a type of phytochrome, a photoreceptor. Okay. So the phytochrome that functions in the ethylation occurs in cytoplasm. Phytochrome functions in light detection during the ethylation. So that's when the first day comes into place. So they are starting to detect the light. Now we go to the transduction pathway. So receptors are sensitive to very weak environmental and chemical signals. So how do these information from these extremely weak signals amplified, and how is the reception transducted in a specific response by the plant? The answer is second messengers. Internally produced chemicals that transfer and amplify the signal from receptor to other proteins that cause the response. In the ethylation, for example, each activated phytochrome molecule may give rise to hundreds of molecules of a second messenger, each of which may lead to the activation of hundreds of molecules of a specific enzyme. By such mechanisms, the second messenger of a signal transduction pathway leads to a rapid amplification of the signal. Now, you may think, does light do to the plant, and what happens there? Okay, so light causes phytochrome to undergo a conformational change that leads to increase in levels of second messenger, cyclic GMP, and calcium. Changes in cGMP levels can lead to ionic changes within the cell by influencing properties of ion channel. Cyclic GMP also activates protein kinases, a diverse class of enzyme that influence activation of other enzymes by phosphorylation. Changes in cytosolic calcium levels also play an important role in phytochrome signal transduction. The concentration of calcium is generally very low in cytosol. But phytochrome activation can open calcium channels and lead to a transient hundredfold increase in cytosolic calcium levels. Like cGMP, cytosolic calcium can influence activation of specific ion channels and protein kinases. So, here is the first day of exposure to light. It's been in front of the light for 24 hours. Its receptors has kind of sensed the light, and it's gonna start going through the pathway, and we'll see the changes. Now this one is the second day of being exposure to light. It's been from light for 48 hours now, and if you look closely, you'll start to see that the branches are turning kind of yellowish green color, and you can see that they're starting to get comfortable with the situation and maybe start photosynthesis or something, because the previous condition, remember, was because it was exposure to darkness. So now that we have put it in front of the light, it's going to go through the pathway as I explained, and so yes, they are turning a little bit green. Whereas this one is the third day. Well, there is not so much of a significant difference from the second day, but still, they're still going to be greener and to be able to make food and photosynthesis. Now on the fourth day, these are becoming more noticeable and you can notice them better. On the fifth one, it's getting even more greener, and it's getting more green. And as we go in the sixth and seventh days, they're getting greener and greener and greener. And finally, on the eighth day, you can definitely see how green is changed since the first day, which was completely white. So now, and of course, if I put it more in front of the light and for more days, it's gonna get even greener. Um, so I hope this was helpful, and so thank you. Bye.